Okay, so chapter 5 takes on a lot of material. It's about the digestive system. Good stuff. Love food. Love to eat. So this is a great one. Um, Also covers plants, animals, bacteria, enzymes that do all kinds of stuff, including break down our food, and energy, this kind of elusive other part of the natural world that we can't actually touch, but it's definitely there and runs everything. Well, this time I want to start our concept map at the end in the physiology part. Let me kind of give a little preview of that. So we got our food, right? Most people kind of make the key mistake that the food gets broken down in the digestive system. That part's right. And then it gets turned into energy. Not there. So the digestive system's job is to break it down first by chewing it up, you know. So we chew it and then we digest it in our stomach, and then it's, in little pieces. But once it gets to our small intestine, then our enzymes get it into really small pieces, tiny, tiny pieces that we can actually get in the bloodstream. All right. And then those get carried around and taken to our cells. That's where the action happens. That's where the energy is released. So we got to start a reaction. See if this works at all. So we got to put a little energy in, and then we get some energy out. We start lighting that stuff on fire, releasing energy into a useful form. Not fire in the cells, it's ATP. It's burning, but you can't really hardly tell. Oh, there's some smoke. There we go. Now, we would actually combust if we released energy this way. So we use enzymes to do it little bits at a time. So let's explore some of that. But first, let's start with the digestive system itself. All right, so here we are on the physiology side of things, and we need to get a little further down. We need to get the food into our mouth, and that goes down, but this drawing kind of disconnects, and so we need to move down here. We got food, makes its way into our stomach, and here we grind it up. We also have a bunch of enzymes that help break that food down. We'll come back to the details of enzymes here in a minute. And then all those little food bits get into our small intestine, and their job is to extract that. This is called absorption, of getting all those little molecules into the bloodstream. And this is a combination of two types of transport: facilitated diffusion and secondary active transport. Some more details on that in a bit. If it does its job right, then that facilitated diffusion and secondary active transport load lots of materials into the bloodstream. Namely, we'll concentrate on glucose and amino acids at this point. And what that's going to do is cause your blood sugar to go up. So now you're going to have high blood sugar. That is going to cause this pancreas here, that's this feather-looking organ, to secrete insulin, because the blood glucose is too high, and that can be unhealthy. So that insulin goes all around your body. There are receptors for it on the surface of the liver. So insulin is received there. It is also received down here on just regular body cells. They have receptors as well. And what they do is install these extra transport proteins that allow the glucose flowing around in the blood to more easily get into each cell. We can think of this in terms of flow down gradients. So blood glucose is high, the cell is low, and we're decreasing resistance to glucose getting in by creating more transport channels. Well, that's what that one does. What do we do here in the liver? Well, we take glucose out of the blood and put it into the liver, but we can't store it easily as glucose. And so insulin makes the liver turn it into glycogen. So if you stick a bunch of glucoses together, you form glycogen. And that's one of those chemical processes that is dehydration synthesis. Let's switch gears and say it's been a little while since you ate and now your blood sugar is low, you're feeling a little maybe lightheaded or you're having trouble thinking. Pancreas to the rescue. Again, there's another hormone it has called glucagon. I know it sounds like this makes glucose be gone, but it doesn't. It does the opposite. In this case, there's just a receptor for glucagon on the liver, and it causes the opposite reaction. It says, hey, remember all that glycogen that you had stored up? I want you to turn it back into glucose, and I want you to release that back into the bloodstream and get this blood sugar back up so I can think again. So there's the whole system. Lots of chopping up the food, facilitated diffusion, secondary active transport. We get into the bloodstream, and then we have to regulate our blood sugar levels. All right, so now let's go back to the front with the chemistry page so we can talk about carbohydrates and proteins. Remember, plants are really rich in a lot of carbohydrates, some of which we can use for energy, others we can't. And animal foods are more rich in proteins, which we use mostly to build more proteins, but sometimes we can use them for energy too. We're going to look at carbohydrates first and then proteins. So we looked at carbohydrates. Plants are rich in carbohydrates. When you look at them, they have carbon, hydrogen, and oxygen. And their name even suggests their ratio of 1 to 2 to 1. Unlike lipids that had very little oxygen, these have an oxygen for every carbon. So that's one way to recognize them. The other one is just the shape. Most of the carbohydrates we look at are a hexagon. They have a little oxygen here in one corner and this little sort of antenna in the other corner. So in terms of recognizing it, there you have that one. Most of the time we deal with glucose. There are other monomers, but we usually deal with that one. What do we use carbohydrates for? I think that's probably fairly obvious. We use it as a ready energy source. We do store it some, but usually this is our ready energy source. When we eat it, we use most of it right away. Some organisms, like plants, use it for their cell wall. Remember, not a cell membrane, but a cell wall. This is cellulose in plants, which we cannot digest. I'll also mention that fungi make theirs out of chitin, which is another type of carbohydrate. It's a polymer. And then we also use it for cell recognition, such as our A, B, O blood groups. On the surface, there are those carbohydrates that help our immune system recognize cells. All right, so there are the carbohydrates. What about the proteins? Key feature when you want to recognize them is they have nitrogen. That's very, very important. And that nitrogen is always with a couple of hydrogens, sometimes a third one as well. So we could even draw another one here, which makes it charged. This is the amino group. Makes sense? They're amino acids. Every amino acid has an amino group. They also have this other group at the other end. This is the carboxyl group. And then in the middle, there's this central carbon and hydrogen. Why am I mentioning that? Well, when you recognize them, there's a lot of carbon, there's hydrogen, there's oxygen, that's all the same. Nitrogen's a new one, so that's important. But there's 20 different amino acids. The only part that is different is this one, the R group. So think of like when you do long division, the remainder. This could be as simple as a hydrogen, or it could be some crazy big ring structure with lots of carbons and all kinds of other stuff. So That's a weird drawing. Kind of looks like Doofenshmirtz. What do you know? Well, that's how to recognize these guys. What do proteins do? What doesn't it do? It varies tremendously. So these form enzymes, they form structural molecules, they transport oxygen as hemoglobin, they form structures like hair and fingernails. Just about any job there is to do, proteins do it. So very, very, very important that we get plenty of protein in our diet. We don't need huge quantities, but enough to make more proteins. If we have plenty, we can use them for energy too, but we mainly need them to reconstruct those proteins that just wear out on a daily basis. Okay, so now on to cell structure and function, where we talk about more membrane transport. Well, here we are in the cell, and we need to talk about how this stuff gets in. So let's go to the bottom of this page where we've done a lot of membrane transport before, and let's talk about this glucose and the amino acids that our digestive system chopped up really nicely for us, got it in our bloodstream, and now it needs to get into the cell. Well, glucose is going to come in, but it has to go through this carrier protein. Amino acids, same thing. They're going to move through those carrier proteins. These are like little sort of doorways. Think about it that way. And glucose, we spend a lot of energy getting it into the blood, and it's fairly high concentration. Presumably the cells are using it up, and so there's low. Same thing with amino acids. There's probably a lot out here and not much inside the cell, so they just want to flow down their gradient. And when they do that, but they need this transport protein, that is facilitated diffusion. All right, so facilitated diffusion, really important. This is going to come up time and time and time again. So same as regular diffusion, high to low is just spreading out, but these need a doorway. Why? Because they're polar. Now, there was another case where we had to get glucose into the cell, but the cell lining the intestine. And in that case, it was a little bit strange. That cell had been packed full of glucose. So it was really high in concentration, and it was relatively low outside the cell. So that seems like, why would it flow in? Well, it wouldn't. So in this case, we're going to need something different. We're going to need to take sodium in, and sodium has a low concentration inside the cell, for reasons we'll talk about later, but relatively high outside of these intestinal lining cells. We can't just shove that through the membrane. We need a membrane protein right here. This type of transport is kind of a funky one. Both of these are moving together. One thing we could call it is a type of co-transport because they're moving together. The more technical name for this is secondary active transport. So that's a special case used primarily when glucose needs to get into the cells lining the intestine. Remember, they leave the other side through facilitated diffusion. One more type of membrane transport to talk about, another type of active transport. So let's say we have calcium inside the cell. Cells don't like a lot of calcium inside, and so there's a special membrane protein, we usually call it a pump, that its job is to get rid of calcium. So calcium concentrations remain low inside the cell, and so they're relatively high outside the cell. Again, kind of like down here, we don't want to do that. Going from low to high, that's going to take work. So in this case, the power for this comes from ATP, which gets made from our mitochondria. So ATP energy gets used, that cranks this pump, calcium gets pumped out, and that is known as primary active transport. All right, so our slate is starting to fill up pretty well with all these different types of membrane transport. We have simple diffusion, facilitated diffusion, and now primary and secondary active transport. Really just a few more to go, but those will come on in later chapters. Okay, so next up is metabolism. This is where the energy comes in and the enzymes. So these are pretty important ideas. We will end up spending a lot of time on metabolism over the course of this class. And this is really about energy. Now, metabolism is really just all the chemical reactions that happen in a cell, but so many of them have to do with energy and transferring that energy. So why do I have a picture of the sun and a leaf? That seems a little strange. It's because that's where it all starts. And this whole idea of moving energy from one place to another is known as thermodynamics. And so the first law of thermodynamics says that we can take energy from the sun and capture it in a plant leaf. If we zoom into the cells, then we can see that ray of sunshine hitting the chloroplasts. They capture that energy in their electrons, and what they do with that is they actually strip an electron off of water. It becomes oxygen, which is a waste product for the plant now, which we need. That's great. But it now has this electron, which it gives to ATP, and it then uses that to take carbon dioxide in and make glucose. So all those energy transfers from the sun to glucose have to do with thermodynamics, saying, yep, we can do that. We can transfer energy from the sun to an electron to one molecule to another molecule to another molecule. And now that energy from sunlight is stored as glucose. Thank you, plants. That was very nice of you. That's kind of their big job as far as we're concerned, is to do that. What we do then is we extract that energy. So we take that energy and transfer it into our chemical reactions to drive the construction of ATP. So we have an adenosine with two phosphates in this third one, and we just pop that right on there. And that has energy. And this then can do work for the body. And then this molecule is broken down again, which means we need to take more energy from more food and power it up again. So that kind of comes about a little bit later. So let's trace this back. In general, we'll go through this in detail in a different chapter, but here's glucose, comes into the cell by facilitated diffusion through that carrier protein, and its energy is slowly extracted step by step by step. Some of it transferred into ATP, some into other molecules. It's then given to the mitochondria, and then extracted into more ATP and other molecules, and so on until we get a bunch of ATP, which then we can use to do work. All right, so there's the basic idea. All that starts with the sun, gets put into our food, and we'll get into this process later on in the mitochondria and how that happens. Now, if you know about the digestive system, one thing you probably have heard plenty about is enzymes. Enzymes help us digest our food, but that's not all they do. So I've separated these two enzyme reactions into the following. There's exergonic reactions. That means energy exits. And there's endergonic chemical reactions where energy enters them. It requires it. All right, so let's start with just this basic process. Most exergonic reactions, we're going to start with a larger molecule. Here's two, let's just call them glucoses linked together. They then fit with the enzyme into the active site, the place on the enzyme that's shaped just right to fit those guys. And then a chemical reaction occurs. It releases energy. Again, energy exits the reaction, and these guys are broken apart. And those are our products. So this particular reaction released energy. And usually that happens when you break things down. I should also go back and mention this we call our substrate. That is the molecule that the enzyme acts on. Now let's look at the chemical reaction and graph it here. This is the amount of energy in those molecules. So lots of energy, high energy at the top and low energy down here. And the reaction, this is kind of like the time progressing from the beginning to the end. And so if we were to watch what happens in this chemical reaction, here we have the substrate with a lot of energy in it. And now we need to add some energy, and this is known as the energy of activation, the energy of activation, or EA. So the reaction can't just start all on its own. Molecules don't just fall apart without reason, so we have to push them a little bit and give it some energy. When we do, they release that energy, and so those products have relatively less energy in the end, so those are our products. But meanwhile, it gives off some energy that we're hoping to harness somewhere in our body, usually in our cells. The things that enzymes do for us is they lower the amount of energy needed to start the reaction. Otherwise, our bodies would just need too much energy to make things happen. And so that's one of the big things the enzymes do, as well as control what happens. You know, what would happen if they cut this molecule up here, but that might be completely useless. Enzymes make sure that it gets cut at exactly the right place so that we get the right products. So that would be a good example of something that would happen in the digestive system. We break down lots of molecules. We break down big carbohydrates into glucose. But we do have endergonic reactions as well. Entering, we're building large molecules. Here you see one here. This is being built. We're going to add this monomer onto it and create a bond. That's going to require energy to be put in. And then we're going to add this one next, and another one, and make a huge molecule. So this is what plants would do with glucose to build their cell walls. Plants would do this to build starch out of glucose. We would do this to build big proteins out of amino acids. So enzymes help that as well. Just like the other one, there's this active site, and that fits the substrate just right. So in this case, this thing right here is the substrate, and the product is this long thing over here. Well, that takes energy. So looking at it here, the substrate that we start with, all these separate pieces, they require the input of lots of energy, probably from our food, and then enzymes make that a little bit easier to happen than otherwise might, and we end up with a lot of them linked together as the final product that is higher in energy than all the little separate pieces we started with. So enzymes can do both. In our digestive system, we focus on the breaking down, but they do the other as well. One last stop, growth and reproduction. Let's talk about bacteria and how they divide. A little different than how we divide. So it's on the same page as mitosis that we talked about for us. Bacteria divide differently by binary fission. With growth and reproduction, if we go to the bottom of this page, then we'll see where the bacteria are in binary fission. In contrast to us, same scale as mitosis, we're looking at micrometers, but this can happen in as little as 20 minutes, not 8 hours. So how is it so fast? Well, it's really simple. So bacteria have this single loop of DNA in the middle, and what they do is DNA replication. So after that's done, they have 2 loops of DNA. The cell then splits apart, and now you have 2 cells. That's really the whole process. So DNA replication comes in heavily here. They have far less DNA than we do to start with, and only one chromosome at that in this big loop. Split the two cells apart, and you're done. So this is how bacteria can divide so quickly. And sometimes that's a good thing, like in our intestines, which are full of helpful bacteria. But in something like a wound that gets infected, this could be a real bad thing.